Hey there. Sorry about that delay. How are you?
I'm doing good. How are you?
I'm good. So what's
Um, so I wanted to just I answered like the rest of the cars question that we went over. I thought I saved the screenshot, but apparently I can't find it anywhere.
okay.
Um, like I thought apparently it didn't save. But I tried answering the rest of the questions, and I wanted to try to get through some of the the biochem from like the content diagnostic because there's some concepts that I'm still
Hmm.
uh can't seem to wrap my head around, like glycolysis specifically.
Okay.
Um and that, so wanted to go over that today. Does that sound like a good plan?
Yep.
So I guess I can sure have the right yeah, I tried trying to make this look like Jack Weston, but apparently since I already did the passage, it's not showing me. It's just going straight to the next passage. But let me just share. Yeah. So this is like the screen because I wanted to go look back to at the passage that we did.
Uh,
I'm not sure how to reset it.
and well, what did you say about the Jack Weston?
Like the like the extension is not popping up. This is like from because I already did the passage or something.
You
I
can
can't
go,
go.
you can go back and I could show you how to do it. You can,
Um
yeah. So just hit open with, or sorry, down review all with Jack Weston.
uh,
Yep.
oh okay. Yeah, I guess like this was like the one that we were doing.
Hmm.
Um so let me just do that. I think we got past question 5. That was where we were at. Um,
Okay. Wow.
um, I decided that. Oh yeah, I think yeah, this is where we were at the last time. We didn't do 5, 6, or 7. But I got a 5 correct.
Okay.
Um, I yeah, what I didn't get right was question 6.
Hmm.
Um, it yeah,
Um,
I think this was like a extrapolation type. Sorry, beyond the text question. It looks like to me.
let's see. After recording technology enabled printed transcriptions of Native American languages, it was noted that stories of contemporary life written in these uh, languages retain the style of traditional oral stories. What is the relevance of this information to the passage? Um, okay, tell me what this question is asking and tell me what your approach was.
So I thought like what this question was asking was I guess I wasn't sure what this question was really asking. I just saw that it was asking about like, hey, they have a re-recording technology and now they're using it to transcribe Native American languages, and it's saying that the language remains unchanged.
Okay.
Like it remained like the same style as it was before. And it's saying, how is this relevant to what we learned in the passage?
Hmm.
Um, and I believe like it was talking about how sometimes society can influence technology or technological determinism, social determinism, and how like the author like believed that nothing like this doesn't happen in like a vacuum, that you can't really say it's technological or social determinism. There's just other factors that also influence the outcomes,
Okay.
whether it's social, technological.
Uh-huh.
Um,
So this is telling us that with the advent of this new recording technology, that the style of these stories did not change. So so that seems like it would go or so it seems like it would go, I guess. So okay, the passage talked about how technological, sorry, technological changes like the type of literature. Sorry. Uh Sorry, I just had someone who I have after you just text me and say that uh if we can reschedule. But okay. Sorry. So back to this. Okay. So we talked about how technology can change the form of literature or like, the expansion of literature. And this question saying that suppose we had this technological change and we applied it to Native American languages, we found that there was not a change. Like the change in technology did not change the type of literature here. So if we look at the choices, so let's see. A says it supports the idea that economic factors are irrelevant to technological developments. What are your thoughts on that one?
so this one, I got rid of that one because the passage didn't really talk
Uh-huh.
about economic factors. Even though like, the author would argue that this could be part of like the situation when it comes to technology, but that's irrelevant to what the question is asking.
Yep. So we could say this is out of scope. Okay.
Um, then like, okay, it supports the idea that technology does not determine the form taken by literature. So this one, I'm leaning more towards right now. I'm just looking back at the answer choices, but um, because like the passage doesn't really say that, that like the technology really like determines the form taken by literature. Like it could, depending on what the technology type is, um, like a printer, for example, you could hit like a book, magazine, or something, or newspaper. It
Did,
doesn't really determine what type of form that you want. I think it's just what the society demands, really.
yeah. Do you remember what we wrote for our main idea?
Like the main idea is that it wasn't that it wasn't only you can't really say that it was technological or social determinism that will determine what is created or the effects on society. That doesn't only happen in a vacuum.
Perfect, perfect. And I like that you were able to, you know, remember that. Because the thing is that one, if you really, really understand the main idea, then you should be able to explain it the way that you just did without having to like, jog your memory. So very good job on that. So yeah, so the passage main argument is that is against, technological determinism and stuff. So B seems to reflect that. But let's take a look at C. So that says it weakens the idea that literacy is necessary for the existence of a market for literature. Now, there's two things that I can maybe predict that you might say, but let's see.
So, the passage didn't really say that like literacy is necessarily like necessary. So it doesn't, but yeah, like literacy, like you do need to kind of read, but like for at the exact same time, there like might be like some pictures or something that people might need want to look at. Like, so I don't think literacy isn't necessary.
So that is what I predicted you were going to say, and you want to be careful about that because like whenever someone says like, they never talked about it in the passage, like it's very, very risky to say that because two things, either it's implied or, not explicitly stated, or it's like some worded in a different way. So we did say that growth of the reading public, right? Growth of the, so, so we said over here, growth of the reading public, right? And then we said new reading public,
Mhm.
and we also said, I mean, thing with like lawyer, okay, yeah, lawyers and merchants that like to read. So yeah, so we said a lot of things in the passage about literacy. So uh
Oh,
mhm.
so is the answer wrong because it would technically strengthen the author's argument that literacy is needed for the market of literature because right now they're just transcribing it and they're making a recording, but it didn't change the fact that they're still orating and being oral, and that's not like literature or
uh so I was thinking about that. Um when I first read the question, I was like, well, when I first read the question, I was like, is this the right passage? Because it was just like it seems like it's like out of nowhere. But I think that it could be, well, while it might be true that literacy is perhaps necessary or at least increased the amount of literacy, it can create that market for like printed texts. Like that is an argument, but I don't think it answers the question of how technology changes, right? But I do see what you're saying about, literacy. I mean, like, you don't need literacy to, like, recite an oral story. But I think the best reason to eliminate C would be that, that this question stems talking about technology and C is not. So, but
Okay.
on the tricky side, yeah.
Mm-hmm.
For D, it weakens the idea that new technologies are responses to demands by the public. Okay, what are your thoughts here?
Okay. So I could see how this answer is incorrect because from our like main idea that sometimes technology can be from the responses from the reading public because we mentioned how sometimes there's a demand from the new reading public.
Mhm.
So the technology might adapt to try to meet it, even though it's not necessarily like a cause, like social or technological determinism.
Yeah, so the passage did talk about new technologies being responses to the demand by the public, but the question stem isn't saying anything like that, right? So so this question's a bit on the tough side because um B, C, and D are all arguments that were made in the passage. I think A is the only one that really isn't. Um, so B, C, and D are like valid arguments, but the one that answers the question stem um would be only, I guess, B. Because yeah, because C, while that is an argument, I guess nothing in the question stem talks about, or I should say less so that, but more so the question stem talks about technology and the style of literature, I guess. So that's what we want.
And what would you exactly say like the question stem is? Because I guess like that's kind of
Oh, so the
confusing to me to figure out what the like looking at the question, like looking at a question and figuring out what the question stem is.
got it. So the question stem is saying that um, technology uh, improved and it enabled um, like Native American languages to be to go from oral to like written or printed. And then it's asking how is this relevant? So maybe I would write something like um, Native American oral to um, literature after tech improved, relevant, or actually, hold on one sec. Literature. I would say this: tech improved, but lit stayed the same. I think that's what the question stem is asking us. And so,
Okay, then B would answer that question stem versus
yep. So you could have even started out by saying, by seeing that we're talking about technology and A and C do not talk about technology. So A and C could be out. And then it's a matter of B. Oh, and then it's a matter of the literature staying the same. And so between B and D, the only one that talks about literature is B. So that could be how to do this.
okay. Thank you. That makes sense. And yeah, and I got a seven correct. So I guess yeah, that was all from oh yeah, and this was a passage that I was doing um the other day
I
when
was
I was trying to practice mapping.
Yep.
I just like noticed that my maps are wordy.
Yeah,
Uh
that's fine because and that's a common thing that students tell me, and I tell them that ultimately it's time. So if it take it if it's wordy but it ta doesn't take that much time to write that, it would take even more time to like try to think how to you know, shorten it or whatever.
yeah, and it did help me not to look back at the passage, except for when I forgot to write down an example that I
Oh, yeah.
saw.
Yep, yep,
And it was in this like big paragraph, but it at least pointed me where to look, so it wasn't like I was just reading down like the paragraph or trying to read the whole entire thing and trying to skim. I'm like, okay, paragraph five, mention this.
yep.
And
Do you want me to look at your map
let me because it's on my I had
and um is there a way you can transfer the file over or maybe take a picture of it and
I
then
could take a screenshot and then send it
yep,
to you.
yeah.
Yeah, because I got a 4 out of 5 on this passage.
Okay. So you
But
probably yeah.
it was like a similar type question to like what I missed on that passage. But let me see if I can
Or you could
take
like read it out or if there's was there like a cards passage that had like more than like a good amount wrong that you did?
not yet. Yeah, I haven't been doing. I only done like one parts passion so far.
Got it, got it.
Because I did have a question about like how I'm mapping.
Yep,
So that I could do more once I get more comfortable with mapping.
got it.
This is like my first time doing it, and it did take me a while,
Yep.
but I'm gonna pay more attention to time the next time I do one. Um, I am trying to send the
Yep.
and I wrote some notes on it from like what I thought like I was missing so far. So I did send you the screenshots.
Let's see.
Maybe I can open it on my end as well, if that would be helpful to do.
All right, so let's see.
Did it come through now?
P1, human diversity, long term and short term and long term plasticity, new environment, for example. All right, so I let me just quickly make a map so I can compare it. I'll try to do it fast. So
Okay.
scientists trying to understand human diversity has to deal with the problem of short-term adaptation or acclimatization and long-term related to survival in a particular environment. We know that individuals possess enough plasticity to adapt to new conditions in the short run, for example, tanning. However, this plasticity is different from the adaptive capacity of individuals living in an environment for a long period of time. So, okay, we have short-term acclimatization. Um,
Work.
okay, um, plasticity, and then long-term adaptive, and they are different. Paragraph two, in humans, other animals, climatic adaptations for genetic. Okay, for different, I mean, good. So they give us a numerical number, so we want to write down which ones those are. So we have genetic, slowest, behavioral, fastest. Genetic is also intergenerational.
Yeah, that's why I added that to my map because I said like a slow and intergenerational.
Hmm. And then we have, okay, growth, which are ontogenetic, which means irreversible within lifetime, and then physio, reversible, and we have an example of like the menstrual stuff, okay, and then P3, behavioral is greater importance than other animals, dealing with stresses, and it's not, okay.
I don't know if you would count although as a contrast word.
Yep, it is.
Okay. And like what about like a how like a contrast word that comes after a semicolon? Like would you count that? Because sometimes I've heard like a semicolon
Oh,
is like two separate sentences.
yeah, just if it starts off like if a sentence starts off with a contrast word, so not for the semicolon.
Okay.
Like it'll still be a contrast word, but it wouldn't be like the type that I would
Write down.
yeah. I mean,
Okay.
if it's important, you write it down, but it's just not what I would like highlight in pink or whatever. Okay, let's see.
Sorry, yeah, that's just what I remember from mapping that I saw, like a
Mhm.
semicolon and a however or some contrast word there. And I was like, would that count? Yeah.
Yeah, I don't know. Uh,
Yeah.
the- I wrote paragraph one, short-term acclimatization plasticity, long-term adaptive, P2 genetic or uh, intergenerational, behavioral fastest, growth, ontogenetic slash irreversible, physio reversible, i.e. menstruation, and then three, behavioral is more important for humans. It's non-genetic, generationally transmitted, helps us in for new environment, and it's also culture. P4, Allen's rule says that, yeah, appendages are shorter in cold climates, and Bergman's that body size is larger in um, cold climates. And then P5, that these rules were relatively unknown until 1950, and it's not uniform. Like it's not like there's going to be exceptions, but it's still pretty useful. So, okay, now let me take a look. So P1, you wrote human diversity, short-term and long-term plasticity, new environment, and then example tanning, and then plasticity, you wrote is not the same as adaptive capacity. So that is, yep, that's good. Okay. And then P2, four adaptations, genetics, slow and intergenerational, behavioral fastest, growth within the individual. And then it says
Irreversible. Yeah.
irreversible. Yep, yep. Physio reversible. Perfect. And then P3, behavioral greater than
It's greater for humans,
yep,
you know, that's, yeah.
perfect. And then Would it?
not genetic. It's intergenerational like
Yep, yep.
transmission, which was from culture, which is why I just put
Got
a
it.
culture.
Good, good.
Um, and it was the most efficient, and you can easily adapt to other environments, which I noticed that was something in P1
Got it.
that mentioned.
And then P4,
Um,
Allen rule, longer limbs hot, shorter limbs cold. Bergman, tall and skinny, warm, short and stocky, cold. P5, author argues that Allen and Bergman, um, what?
can
Huh?
um, so I guess like for for this one, I think I was slightly off on what the author like I kind of misunderstood it. I think I was trying to write that the author argues that Alan Bergman can like I think I misunderstood it. Kind of was thinking that Alan Bergman can explain the adaptive traits,
Huh?
but obviously in the paragraph that it was mentioned that not the scientific community doesn't agree with that. Um
Uh-huh.
so I think I missed the point on paragraph 5 now looking at it.
Uh,
Um
okay, you know what I think actually now? So is that however at the end? Well, actually never mind. But yeah, I think that's just saying that um that it's not like even now no one argues that these rules can be strictly applied to animal populations in all cases. So that's probably the only one that you didn't write, but everything else is perfect.
Yeah, I just realized, yeah, I just messed up on paragraph 5, and that's probably why I got the question wrong.
Which one? Show me the question.
Um, let me go back and see it. 9 I got right, 10 I got right, 11 I got right, 12 I got right. Or maybe what was that question that I changed? Did I change my answers by accident? Oh, this one was the one that I got wrong. So originally I think the answer that I chose was C,
Okay.
but this is because I just jumped the gun relatively quickly. I didn't really understand what paragraph 5 was about, I guess.
Mhm.
But originally I chose C. But it's an example of you see it in the passage, but it didn't answer the question.
Oh, yeah, yeah, yeah.
Yeah.
So,
Um,
and also, though, is it like Allen and Bergman's fault that people didn't really know that much about it?
no.
No, right?
Yeah.
Um, but but yeah, so like yeah, you can see how this could be like the trap answer choice because it's explicitly from the passage. So you want to yeah, just be careful about that. And for the other ones, A were formulated in the 19th century. That's true. But that's not really like a
Yeah.
drawback. And then have little explanatory power. I mean, that could be a drawback, but they are saying that, you know, like they are possible explanatory models for various adaptive traits. So the author is saying that they do have some good explanatory power. But yeah, where it says no one, argues that these rules can be strictly applied to animal populations in all cases. So that actually is using words from the passage too. Um, right? Because it says,
Hmm.
you know,
But no one
apply.
can. Oh yeah, because there's like no one will argue that the rules can strictly be applied, and that's why I highlighted this sentence here.
Yeah. So for the highlights, like you highlighted argues that these rules can be strictly applied, I would have highlighted um, no one or no
Yeah,
applied all cases. So,
and
but
that's probably why I missed it because I just completely.
yeah, yeah. But I mean, you still did a great job. Your map was pretty much perfect, and your highlights are really good. And yeah, it seems like you really understood it. So great job.
Yeah, again, just gonna we'll see if that continues. But with other practice, I think I just need to practice and do more. Um, and then yes, I'll just come back if I have difficulties and if I'm still missing those types or certain types of questions that. But the map really does help,
Yep.
like when it comes to not because like almost every time I had to go look into the passage, I got the
Yep.
answer wrong because I see something that
Yeah.
remotely resembles.
Yep.
And then I'm like, I'm done when not really.
Yeah. That's how most people uh do cars. And I remember like when I was first teaching it, like before I blocked the passage out, like I could see like my students' eyes just frantically searching through the passage and playing mix and match. And yeah, you don't want that. So yeah, excellent job. And you wanted to show me some stuff about
Bye.
like the sciences now?
Yeah, the sciences. Let me and I think it was this one. Um, yeah, I did have a quick question before we talk about like glycolysis. Um,
Say that again?
I have a quick question before you know talking about glycolysis because those were the types of questions that I did,
Got it.
and uh, and I understand inhibition and competitive and all that. I gone over that myself. And that was not bad, but
Okay.
I think it was something about the amino acid weight. Will I have to know for the MCAT that the average weight for an amino acid is
Yeah.
110 Daltons?
Yeah.
Okay, because like this is something I just didn't know. So
Yeah. So I'm gonna show you like for the science passages to look out for words I call buzzwords, which are words that they'll likely ask you questions about. So if you ever see something like, it's a 552 amino acid, enzyme or whatever, expect to be asked how many kilodaltons that is or something.
okay,
Definitely.
okay.
Yep.
Let me see. I think it was 2029 is where it was just at. Hopefully. So I guess like I don't know like why it's hard for me to wrap my mind on around like what happens during glycolysis because is it like I'm pretty sure like the process of glycolysis is to create acetyl-CoA in
Uh,
order to be used in the Krebs cycle and fatty acid synthesis. 'Cause I
so
think I was, yeah.
yeah, so the thing is that um all so what's catabolism?
Catabolism is to break down.
Uh-huh. So so when you break down the biomolecules like sugars, fats, proteins, they will at some stage in their catabolism will turn into acetyl-CoA. That's where what they all have in common. And acetyl-CoA, if you have oxygen, can enter into the mitochondrial matrix and undergo the Krebs cycle, which then causes the electron transport chain to happen. And that's where you get, your 36, 38 ATP equivalents. But glycolysis is the breakdown of glucose into pyruvate.
Okay.
Okay. Now, um, so I shared that Google Drive with you, right?
Um, yes, I should be able to find it. I remember trying to look at it somewhat. Shared with me and cat. There we go. And then it was under, where did I find it under? Oh,
Oh,
PDS.
you could share your screen and I could walk you through it.
Oh, it's not the sharing. Oh, I didn't realize.
Yeah,
Let me,
it's just on that. Yeah.
yeah. Okay, I wasn't realizing that was not sharing. Um, just hopefully it's sharing the right, like the right one. Do you see this one?
Yep. So yeah,
It
hit
was
yeah, and you're gonna see a PDF called Metabolism OneNote. Hmm.
one note.
Yep.
Doesn't matter which one.
Oh yeah, they're the same one.
Yeah, okay.
So here, it's weird that it's showing in such bad quality.
Yeah, what is this? Like, where should I be going? I don't know. Yeah.
Let
Mm, oh,
let's try. So weird. Let's try this. Let's move your mouse up towards the top and hit that download button here. See where
yeah, it's downloading.
yeah.
Yeah.
Let's see if yeah, and then let's open it up that way.
Yeah. Can you still see my screen or do I need to share it again?
I could see it. But that's
Okay.
weird. Like, is it showing up blurry for you?
It's still kind of like, I mean, like when I like zoom in, it's not as blurry.
Oh, it's fine. Okay.
Yeah, so it's not blurry. Yeah, I
Got it.
think it's just too small.
Got it. Okay. So yeah,
Yeah.
when so like yeah, when I was writing this out, like I noticed that like I couldn't find like a source where they put all the pathways in the same cell. So that's why I made this. And I wrote it on OneNote. I don't know if you're familiar with that, but it's like it's supposed to be this really big canvas, but
Oh,
I had to export it as a PDF, so it's cut off. But what isn't cut off is this glycolysis. So on the left, you can see is glycolysis. So basically we have like um glucose entering into the cell and the teal colored steps are the irreversible steps, which means that they have a delta G less than zero, which means spontaneous, right? So this first step here, glucose. Um, so what's happening is, well, yeah, what's happening to the glucose in this first step?
You're talking about like yeah, the glucose that's outside of the cell.
Oh, when it's inside, like,
Like when it's inside. Oh, this one. Um, so it seems that hexokinase
huh?
is going to tack on a phosphate
Uh-huh.
onto glucose
Good.
to make it a G6P,
Good.
and that phosphorylation step is to keep the glucose inside the cell so it can't leave.
Yeah, yeah, yeah. Did you see that in one of my other PDFs, or you just,
I like it was like when I was trying to learn about like
yeah.
glycolysis. I think it's just like for me like that is like how much I understand. After that, it's
So that's good
well,
because a lot of people don't understand the why, they just understand what it is. But also a really high yield topic is kinase, which is a type. So like you need to know like the different enzyme classes, and the kinase is a transferase, and that just means that it transfers like a phosphate group. So it could take a phosphate from ATP and give it, in this case, to glucose to make glucose 6 phosphate, and then you're left with ADP. But it could also do the opposite. So now, in addition to that, you're expected to know the um that kinases use three specific amino acids, and they are S, T, and Y. What are these?
Y is tyrosine. Um, T is threonine,
Good.
and S is serine.
All right. Now, what do these all have in common?
Um, like okay, I know like serine and threonine, they have the hydroxyl group attached.
They were good.
Um, Tyrosine, I guess tyrosine also has an OH, but I don't remember seeing it on that, but maybe it
Yeah.
does.
So the,
Yeah.
the tyrosine will have a benzene and then an OH attached to the benzene. And now I want you to like train yourself to whenever you see an OH group to think nucleophile. Okay?
Okay.
So I'm gonna draw like a squiggle and then an OH group here. And that is the most common nucleophile on the test. The most common electrophile is going to be some carbonyl group like this. And that's because a carbonyl group has the dipole, which means that the oxygen has a partial negative and the carbon has a partial positive, right?
Mm-hmm.
And nucleophiles, like in the name, they like nuclei, and nuclei are positive, right?
Mm-hmm.
So this is going to attack the partial positive carbon here. And since carbon can only make four bonds, one of these bonds, double bonds, has to go to the oxygen, and then you would get something like O minus, the Lg, and then the OH that is attached to like the squiggle. That's your like intermediate here. But you know, oxygen doesn't want to have that negative charge. I mean, like it does, but it's not as stable. So it's going to recreate that double bond. And since carbon can only make four bonds, one has to leave, and that's what the Lg is. It's a leaving group. So this leaves. And then you're left with, you know, like this kind of deal. Okay. So now applying that same concept to ATP would be like this. It would be like, so like, let's say this box is like the adenine or, the ribose sugar attached to the adenine. And let me actually make it closer here. Um, so first of all, a phosphate group looks like this. And here's another. And here's another. And then let's say this is the adenine. So now this is ATP, what I drew. And by convention, as we move outward from the adenine, like the closest one is going to be alpha, followed by beta, followed by gamma. Now, just like the carbonyl group has a partial positive because of the double bond to oxygen, the phosphate, or rather, the phosphorus atom also has a double bonded O, but it also has three other oxygens. So it's going to be very partial positive here, right? And for the same reason that the OH would attack the carbonyl, it'll attack the phosphorus here. And it'll push this double bond up here, but then it'll come back and it'll break this off as a leaving group. And so that's how you can take, or that's how a kinase can use ST or Y to remove the gamma phosphate of ATP to give it to like the substrate. So that's what's happening here. Because you start off with the ATP, but it rips off the gamma phosphate and gives it to the glucose, and that's why you're left with ADP and a
Mhm.
phosphorylated glucose.
Mhm.
Okay. So expect to see lots of questions about this. So connect in your head. Um, Kinase, it's a transferase, and it transfers phosphates, and it uses STY, which have hydroxyl groups, which are nucleophiles, and a phosphate is an electrophile, and so the STY can nucleophilically attack the phosphate, outermost phosphate, rip it off, and give it to a substrate, in this case, glucose. But it can also do the reverse. It can remove a phosphate from a substrate and give it to ADP to turn it back into ATP. So, any questions there?
Okay. S- So yeah, just to clarify.
Mhm.
So ST and Y, they are nucleophiles.
Yeah.
And then the phosphate, the gamma phosphate
Yep.
of ATP is an electrophile. So can a kinase also attack ADP? And would that be considered the still be the beta phosphate? And can it just change ADP into AMP or no?
Uh, that's actually a good question. Let me see. Okay. Uh, no is the answer. And and if you did want to convert ADP into AMP, you could use, so like the transferase just transfers A phosphorylase adds and a phosphatase removes. So if you want to go from ADP to AMP, you would have to use a phosphatase, but the transferase, the kinase, does not do that.
Okay.
But that was a really good question. Very, very nice.
Yeah, just trying to think it through.
Uh,
And then
go on.
oh no, I forgot.
Any other questions?
Um, the gammas and I guess like my question is, it's called G6 is because there's six carbons, like
Yes.
G6B.
So that's another thing. So you're going to be expected to know how to draw the structure of glucose and the fact that it's could be a straight chain form and then a ring form. So I'll start off with the ring form. So up here we have a like an aldehyde, and then we have OH and H here, OH and H here, OH, H there, OH, H there, and then CH2OH. So something that helps me remember this is middle finger because
That's your right hand.
you could see, right?
Hmm.
Like, oops. So like one, like an OH group on this side, and then on the opposite side, and then this side and this side. You see like OH groups here, and then it's on the opposite side, and it's back there and back there.
Yes.
Make sense? Okay. So that's the straight chain form of glucose. Now, in actuality, around 0.1% of glucose in our bodies exists as the straight chain. So what happens, so its ring structure is going to be much more prevalent. And if you just apply what we already know about hydroxyl groups being nucleophiles and carbonyl groups being electrophiles, you will see that this, so like here, I'll label the carbon. So we have carbon 1, 2, 3, 4, 5, and 6. So what happens is the fifth hydroxyl will nucleophilically attack that first carbonyl group. And that will create, or it'll close the ring, and it will create this type of ring here. And this, I'm going to label this hydroxyl oxygen with orange to show that that is this one as it closes its ring. Now, um, now there's something called like anomers, and what that means is because, okay, the anomeric carbon is this one over here. because that's the one that um can change, something called mutarotation. Now I didn't draw like the OHs here, um, but there would be OHs. Like for instance, if this was an OH down there, OH down here, oops, OH here, OH there, and then this one, OH like that. So if the and so this is the one, I'll put it in orange also. This is the anomeric carbon, and this can change. Like this can have this OH on the bottom, it can have it on the top. And if it has it on the bottom relative to this sixth or the last hydroxyl, if it's on the bottom or like an opposite side, it's going to be considered the alpha anomer, and that's around 34 or 36% um, of the glucose in our bodies exists in this state. But if it was, maybe I'll use green. Let's say that the oops. Let's say that the OH is on the top, right? That's going to be the beta anomer, and that's like 64% of the glucose. And that means that the beta anomer is a lot more stable. And that's because if you were to draw it in the chair configuration, all of the hydroxyls in the beta anomer will be in the equatorial position, which means that they're furthest away. Whereas for the alpha, you'll have them all in the equatorial except for the anomer, which is going to be axial. But anyway, so that's just the some stuff about glucose that you're expected to know. And so yeah, to answer your question, yep, glucose has 6 carbons, and hexokinase seems to phosphorylate the sixth position. And also, so you mentioned like it traps it in the cell. Why would it like how like yeah, why would it be trapped in the cell after you phosphorylate it?
Is it I think I might be recalling this incorrectly. Is it because the glucose becomes negatively
Yep,
charged and that's what keeps it inside the cell?
perfect, perfect. Yeah. Because the things that can travel through the membrane are gonna have to be like nonpolar, right?
Hmm.
Phobic stuff. So yeah, if we impart a charge on the glucose, yep, it can't leave the cell. And then we see for the next step, glucose 6 phosphate turns into fructose 6 phosphate through this isomerase. And an isomerase is just an enzyme that moves stuff around. It doesn't add, it doesn't remove. So that's just a reversible step that we don't really care much about.
Is
And
it kind of like isomers?
yep,
That's okay.
because the iso part means like the same, right? So
Hmm.
we just move things around to make it fructose 6 phosphate. But then we have our second irreversible step, which is caused by phosphofructokinase, and it involves us phosphorylating it again to get fructose 1,6 bisphosphate. All right, and then we have some more reactions with things happening. And you can see that this NAD plus into NADH means that, what does that mean?
So and look, NAD is like an electron carrier, so it got what's the word for it? Because it isn't accepting a proton or like a hydrogen atom, it's getting oxidized.
Oh,
So, or no.
which one are you saying is getting oxidized?
Like NAD plus is getting oxidized to change into NADH.
Oh, okay. I see why
Yeah.
you're saying that. So the thing is, and that kind of confused me at first too. is that this H, so it would be more accurate for me to say like plus H plus, but this H here, so okay, if we have NAD plus turning into NADH, the H seems to have gotten rid of that positive charge, right?
Is it because it's an electron that's getting added to it?
Yep, and and the reason for that is that the you're adding the hydride ion, which is hydrogen with two electrons, which gives it a negative charge. So, because it could be kind of confusing because we typically talk about protons
Mm-hmm.
or regular hydrogen, but you can also have negatively charged hydrogen, which is called hydride.
Mm-hmm.
And so, and that, making it complete would also have me add like an H plus group here just to make the signs equal on both sides. But yeah, the H is a hydride, which is means that it has extra electrons. So it's actually getting reduced. But the oh, and then I should say this, catabolic processes tend to be oxidative, meaning, you know, we're oxidizing, in this case, the glucose 3 phosphate. We are oxidizing that into the 1,3, I think, bisphosphobutyrate or something like that. Um, so yeah, so we're oxidizing like the actual stuff in the actual like metabolic, molecules. Um, and also if you think about how NAD and that kind of stuff are electron carriers, it would make sense that they would gain electrons, right?
Mm-hmm.
So they gain electrons to ultimately give to oxygen at the very, very end. Uh,
Mm-hmm.
yep, so that's what you see here. And the enzyme is, I wrote DH, like gap DH, but the DH just means dehydrogenase.
And I have a quick question. So going back to like the fructose 1,6 bisphosphate.
Yep.
Um, so going before that, the reason why it's G6P is that the phosphate group is being added to the sixth carbon atom. Sorry,
Uh,
not the
when you said the G6P,
yeah, 'cause it's saying the phosphate is it attached to the sixth carbon because I'm trying to make sense for the
oh,
fructose 1,6. Is
yeah,
it the
yeah,
first and the sixth carbon?
yes, yes, yes. Yep.
That's why it's named that way.
Hmm.
Okay.
Yep.
Thank you. Like that was just hanging out in my head for a while. Like, why is it being named in this way? Yeah, it just sounds random to me.
Yep, yep. Yeah. So yeah, the number reflects the number of carbon that has the phosphate. So okay. So we have our 1,3-BPG, and then that turns into some more stuff. And we ultimately end up with PEP, and that just means phosphoenol pyruvate. And now this is an example of, so pyruvate kinase is the enzyme here, but this time we are starting with something with a phosphate and ending with a pyruvate that doesn't have a phosphate.
Mm.
And then we start with an ADP and we end up with an ATP. So unlike those previous two steps with kinases, this is showing you that it can do the other thing, do it the other way around. It can take, in this case, it takes the phosphate from the substrate and gives it back to ADP to make ATP.
And that's why they say glycolysis can make some ATP, but just not as much as the electron transport chain.
Correct. Yeah. So that's going to be like, I think like the, oh, like here and here. Um, are uh, where it makes ATP. But also here, I guess I didn't say this, but here, if we go from 1,3 to just 3, we are removing a phosphate too, right?
Mm-hmm.
So, all right, cool. So yes, the pyruvate is what you end up with, and technically, um, it's 2 pyruvate because a pyruvate is a 3 carbon molecule, whereas glucose is a 6. So, so yep, once you reach pyruvate, glycolysis is done. And then what can happen? Well, if there's oxygen, it can enter into the mitochondrial matrix and undergo Krebs and then etc. But if there isn't oxygen present, it'll undergo anaerobic fermentation. So the pyruvate through the LDH, which is a dehydrogenase, which again means that, it's a oxidoreductase, so it makes like redox happen. Um, but this time, instead of having NAD plus turn into NADH, we have NADH turn into NAD plus. And you might be wondering why. Why is that happening? Like, why did we make NADH just to turn it back into NAD plus? Well, in order for glycolysis to proceed, we need NAD plus, right? So in this fermentation step, we are just replenishing the supply of NAD plus, so it can, be able to undergo uh, glycolysis again. Um, but we end up with lactate then, and the lactate can then travel through our blood to enter into our liver, in which it'll turn back into pyruvate, but it will undergo gluconeogenesis to make from that pyruvate to make glucose again. And
So
yep.
why was it just pyruvate and then changed to lactate, then back to pyruvate? Is it just the way that it travels in the
So two reasons. So one, this is happening when there's no chance for a Krebs cycle. So the only thing that you can do is just make more is to have more glycolysis. And so glycolysis we saw had as its inputs, and if you scroll up, I also wrote like the overall kind of formula. Yeah, so glycolysis here involves glucose plus 2 ATP, causing resulting in 2 pyruvate, 4 ATP, 2 NADHs, and 2 H2Os. Oh, I should have written plus 2 NAD pluses. So I guess I forgot to put that. But that basically means that in order for glycolysis to proceed, you need NAD plus. And like when glycolysis first proceeded here and we ended up with our pyruvates, that's when it's like, okay, do we have oxygen or not? So if you don't have oxygen, it's like, well, how are we going to make energy? We just have to do more glycolysis. So what do we need to do more glycolysis? We need NAD plus, and that's why when pyruvate turns into lactate, uh, it creates NAD plus, right? Which can help with glycolysis. But in addition to that, we also need glucose for glycolysis, right? So that's when it travels to the blood, through the blood into the liver, and in the liver, it will cause gluconeogenesis, which is making taking the pyruvate and turning it back into glucose. And that gives us the two ingredients, the glucose and the NAD plus that we need for glycolysis. Yeah.
Oh, okay. So the point of it turning to lactase to make sure that there is good enough of, or to turn it back into NAD plus
Yep.
in
To
order to.
Yep. So,
have glycolysis instead of just making the pyruvate go straight to glucose.
yep.
But if there's no NAD pluses, it's not going to do anything.
Exactly. Yep.
Mm.
And I think I wrote down there, if you scroll down some, yeah, I put these ratios. So like this ratio, like NAD plus to NADH, like if that's, you know, a high ratio, meaning if NAD plus concentration is higher than NADH, it's indication of starvation. Um, because you're not, uh, you're not like, well, you have, a low amount of the NAD plus and sorry, you have a high amount of the NAD plus, and we know that, if you had glycolysis and stuff happen, you would have a lower amount of NAD plus compared to NADH. And then I also wrote NADH to NAD plus is proportional to feeding, like if you have just fed, if you've just eaten. But yeah, for the most part, that's your um and then this is called, I think, the Cori cycle.
Oh,
Yeah, it's the put an image in the chat.
let's see.
And um
Oh,
oh yeah, if you I think you're sharing just like the window, but if you share the entire your entire screen, then I can see like the thing that
oh, let me. How do I do it there? Share. Um, it's
yep,
giving me screens,
yeah,
but
yep. You could just hit the screen.
oh, I guess you can see that. Can you see?
Yep. Awesome. Huh. Edit your images using AI.
Yeah.
Guys,
Um, oh, where did it just go? Go back. There we go.
yeah.
Okay.
So here you can see that, for instance, in muscle. Oh, and I should have also kind of like showed you in that PDF that um the transporters, like when glucose enters through like the transporters, like there could be like one in adipose tissue. And so like if you just move this over a little to the right,
Um,
huh? Yep, just like hold on to it and then drag it. Yeah.
oh, where did it just
Okay.
uh
Then on the left, if you scroll up a bit,
what? Oh, another picture on my oh, whatever.
yep. Here we see that there's the glut 2, which is in the liver and pancreas, and the glut 4, which is in adipose tissue and muscle tissue.
Hmm.
And so, yeah. So now if we go back to that image, so you can see that in the muscle here, like that's where this glycolysis happens. So we can see that it causes two pyruvates to be made from one glucose, and that makes two ATPs. And, in the case of not having enough oxygen, the two pyruvates turn into two lactate. And another reason it turns into lactate is because lactate is soluble in the blood. Like lactic acid buildup if you like do an exercise like for, million reps or whatever.
Hmm.
Yep. So it turns into the lactate, which can travel through the blood to the liver, where it turns back into pyruvate and then undergoes gluconeogenesis to recreate glucose, which can then go back through the blood to the muscle, and then that can be broken down through glycolysis to give you the two ATPs again, and that's that cycle.
Okay.
Mhm. Yep. And now do you want now let's go over like if there was oxygen. So if you can click back on that OneNote PDF here. So
Okay,
yep.
there we go.
So now if we do have oxygen, the pyruvate can enter into the inner mitochondrial matrix. You can scroll down a bit more. Yep. So you see our pyruvate over here, right?
Mm-hmm.
Okay. Now this thing in like magenta that I wrote is called the pyruvate dehydrogenase complex. And it's like it involves three subunits with three different enzymes. And ultimately, what happens here is, well, you reduce NAD plus into NADH, so you make some of that. You lose a carbon in the form of CO2. So if pyruvate has three carbons, we lose one of them. And we also add a CoA. So that's what gets you acetyl CoA. The acetyl just means you have two carbons, and the CoA is just that CoA. So we're entering into the Krebs cycle. You can scroll down some more. So you can see that we are entering into the Krebs cycle, and well, so we have so okay, I'll kind of write it like this. So we have our um okay, so I'll just say that the oxaloacetate has four carbons, and the acetyl CoA enters like on top of that four carbon oxaloacetate to make a six carbon citrate, which can then just isomerize into isocitrate. So we don't worry about that. In general, we're going to be more so concerned with irreversible steps, right? So reversible step, don't care that much about it. You get isocitrate. And then we have an irreversible step in which we have a dehydrogenase. So we have oxidoreductase. So we have NAD plus getting reduced and the isocitrate getting oxidized. And we also are losing a carbon here. So now we have five carbons. Oops. This is still six carbons, but after we lose the CO2, it's a five carbon alpha ketoglutarate. Um, and then we have another dehydrogenase, which again makes, NADH. And also we are losing another carbon in the form of CO2. So now we have four carbon succinyl CoA. And then succinyl CoA um, well, involves an enzyme that's now a synthetase, which means that it makes ATP, or technically here, I guess GTP, but that GTP can turn into ATP. So that's what's happening there. And you end up with succinate, and it's still going to be four carbons. And now for the succinate dehydrogenase now reduces the FAD into FADH2. And I wrote like in parentheses complex 2 here because this is like directly connected to that complex in the electron transport chain, which you'll see in a bit. Um, but yeah, you end up with fumarate, which again is going to still be four carbons, and then you can add H2O to it to and fumarase to turn it into L-malate, again, four carbons. And then the malate through malate dehydrogenase can be oxidized into the oxaloacetate and produce NADHs. So we still have more, electron carriers being made. And that's the Krebs cycle. And if you ever wondered like what a hangover is, it's so like alcohol inhibits antidiuretic hormone, and antidiuretic hormone in the kidneys, like you can remember it by the mnemonic aquaporins drilling holes. But what that means is that it opens up aquaporins in the collecting duct of the nephron, which means that water gets reabsorbed into the blood rather than urinated out. But if that enzyme is inhibited, it means that the water doesn't get to be reabsorbed and it gets urinated out, and therefore you become dehydrated from drinking alcohol. And the hangover is so our brains can only use glucose for energy. I mean, it could also use ketone bodies, but just glucose aside from that. So if you have if you're breaking down glucose in the Krebs cycle, you can see that you need water to do that. So if you don't have water, then it, hurts your brain and stuff. So that's that's that. So that's that for the Krebs cycle. What are your questions here?
Okay. So do I have to like memorize the enzymes that create like one to like another, like the ketoglutarate,
Yeah, so
the
I would say to understand the enzyme class. So, like, if you don't remember isocitrate dehydrogenase or alpha ketoglutarate dehydrogenase, at least remember that these are dehydrogenases. And so, yeah, I would say that the enzyme itself is less important than the enzyme class, but the most important will be the irreversible steps and then the things that happen there, meaning um, like for the dehydrogenases, you're making NADHs, and some of them you're losing CO2, and some of them you're making ATP, some of them you're making FADH2. So yeah, those types of things, and also the number of carbons can help.
okay. So in the ones in the blue you said are the irreversible
Yeah, the
steps?
ones that so like if the ones where I'd write like a double-ended arrow like this are reversible, and so the ones that are like just a single directed arrow is the irreversible step. Yep. Uh,
Okay.
and you are it's annoying, but you are expected to know like each of these names because I've seen like it's not going to be maybe like a big topic, but like I've seen practice tests where they asked like in one question like what comes after succinate or something. So that could be kind of annoying, but yep, so that's that. And now if we go up,
Oh, there you go. Oh,
oh yeah, just above, because now I'm just gonna do the electron transport. So just like this region, yeah,
right
over
over
here.
here. Okay.
Yep. So we could see here that complex one over here. So this is where the mobile electron carriers like NADH and FADH2 drop off their electrons. So complex one here. So it's a little interesting how it's kind of like arrayed because you have complex one, complex three, complex two, complex four, but you'll see how it makes sense. So like complex one over here is where NADH drops off its electrons, which means NADH is getting oxidized now back into NAD plus. And these electrons are going to this UQ, which is ubiquinone. Um, and as that happens, it pushes four protons out from the matrix into the intermembrane space. I wrote that in red. You see that?
Mm-hmm.
Okay. So the electrons go to the UQ here. Now, remember when I was saying about that succinate producing that FADH? That's directly gonna affect complex 2 over here. So you can see succinate turns into fumarate, and that means FAD turns into FADH2. But when FADH2 gives off its electrons to complex 2, it turns back into FAD. And you can see in complex 2, the electrons go to ubiquinone again. And, maybe I forgot to draw like the protons being pumped out, but that would happen. And so you end up building a pool of reduced UQ. Um, so that means that um, like so UQ is ubiquinone, and it gets reduced into as it gets electrons, but then the UQ gives their electrons to complex 3, which is in the middle there, and you see we're pumping protons out as well. And the complex 3 gives its electrons to cytochrome C, which when it gets its electrons, it gets reduced. It can then travel to complex 4 on the right and give its electrons to complex 4. And after that, it'll just go back to complex 3 and get more electrons. But yeah, it gives it to complex 4. And complex 4 is that final destination, because that's where it gives those electrons ultimately to oxygen. And that's why you have and that's where all the electrons ultimately end up. And so you can see, like, I wrote like half O2 plus 2H plus yields water. And that's because that's what you see at the end of cellular respiration, right?
Mm-hmm.
Like you see the like glucose plus CO2, or sorry, glucose plus O2 yields CO2 and water. So you could see like glucose, we started with that with black in glycolysis, and then oxygen here we're adding as a reactant, and then water here we're getting as a product. And in those Krebs cycle steps, you see that you also make CO2, so that's also a product, and that's the entirety of the cellular respiration.
Oh, yeah, and like is it like the oxygen kind of like electron, sorry, oxygen is the final electron acceptor and it goes through ATP synthase, or is it something else? Because
Oh,
like,
I forgot about that.
yeah.
So, okay, yeah. So all of this stuff basically um creates a high concentration of hydrogen or of protons in the intramembrane space compared to the mitochondrial matrix. And so it will want to go down, its concentration gradient from high to low. And that happens, that's like diffusion without requiring energy. And then over here, we have our ATP synthase, which harnesses that energy of the protons coming down into the matrix and uses that to make ATP. And that is where you get the majority of the ATP from cellular respiration. And something that they like to ask is something like, what if we, the inner mitochondrial matrix, I'm sorry, the inner mitochondrial membrane, right? Like, this one. What if we increase the permeability of that? In other words, meaning,
Like,
yeah.
like increase the permeability of, wouldn't it cause it's like the hydrogens to just simply come down on their own. They don't need the channel.
Yep. Yeah. Smart. So that uncouples the ATP synthase from the, proton gradient. So you're going to end up with like ATP is not being produced. Um, so yeah, great job. They like to ask that question. Like they like to ask like 2,4-DNP is something. It's like a benzene with like two like nitrites to it. Um, but they could also ask like if they ask about like brown fat or like thermogenesis, that could also be something. Because that would mean that the energy can be dissipated in the form of heat rather than being harnessed to make ATP. But yeah, great job. So yep, that's that's pretty much that. So what do you think? Yeah, and then I also have like other stuff. So like for
Okay.
instance, yeah, that one's going into gluconeogenesis, and then this one's going into different types of fatty acid oxidation, and then this is the rest of the gluconeogenesis. So this also highlights the importance of location in terms of, because for instance, we know from glycolysis that glucose turns into glucose 6 phosphate, right?
Hmm.
And then here, this is gluconeogenesis, by the way. Here we have glucose 6 phosphate turn into glucose. So what's up with that, right? Like if you're turning it into glucose 6 phosphate and then you turn it back into glucose, like what happened? Like yeah, what's up with that?
Mm.
So this is called a futile cycle. And you know the word futile?
I mean, isn't that like pointless,
Yeah.
futile? Yeah, yeah.
So this is called a futile cycle, and that would be, yeah, like pointless. So why does our body work? Why isn't it just completely not doing anything? That's where the location comes in because in it's hard to see, but I wrote here in yellow, in the endoplasmic reticulum, there exists this glucose 6 phosphatase enzyme that dephosphorylates glucose 6 phosphate to create glucose. Whereas glucose to glucose 6 phosphate in glycolysis is in the cytosol. Okay. So that's why you don't have a futile cycle because if you're in the cytosol, it's going to be glucose to glucose 6 phosphate. If you're in the endoplasmic reticulum, it'll be like the endoplasmic reticulum simply will have the enzyme that will enable glucose 6 phosphate to turn back into glucose, but it's not available uh, in the cytosol. So that's why you don't get a futile cycle, and that's why um, compartmentalization is important and location is important. So,
Okay, yeah, that makes sense.
mhm.
Um, yeah. Oh,
Oh, yeah,
what's all this?
the pentose phosphate pathway. Um,
Yeah, I think we can go over the like some of the other pathways next time. 'Cause I think I need to just get in the hang of like glycolysis and the Krebs cycle.
yeah, 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 totally.
It could,
Um, did now you have some stuff from the content diagnostic?
yeah, let me see like if it's gonna match any of that.
Oh,
You
yeah.
see this? Yeah, because it
Got it.
says yeah,
Yeah.
so like during glycolysis, the reaction catalyzed by isomerase dehap to glyceraldehyde 3P. Isn't that just G3P?
Mhm.
Um, has a slightly positive charge in Gibbs free energy. However, the reaction still tends to proceed under physiological conditions. This is most likely because
So
uh,
so all of those irreversible steps have a delta G of that's negative. So this is saying that this step has a positive delta G, which means it's not irreversible. It means that it is reversible. But they're asking if it's reversible, why does it still go forward? And we can, so like for A, what do you think about that?
um well it's saying that like ATP breakdown. Um so I guess mm,
So,
I'm not sure. Yeah.
so if it was coupled to ATP hydrolysis, what type of enzyme could it be?
It could be no, but like a kinase is like a transferase, but it does break down ATP, but I'm not sure if it's like specifically hydrolysis. So,
Uh-huh. Yeah. So we can kind of do it in like two ways. So like first, um, which enzymes are related to ATP? You'll have your, transferases, or your, which are kinases. What other ones could there be?
I forgot like which enzyme class is like a phosphatase, but
So that would remove a phosphate, and so phosphatase, and then if you added a phosphate, it'll be phosphorylase.
mm-hmm,
So right away, that shows us that if we're dealing with ATP at all, we should see those types of enzymes, right?
mm-hmm,
You saw the hydrolysis part, which is just making it even more specific because now you know that that would be a phosphatase. But if you didn't know that, at least you know you're looking for a kinase, a phosphatase, or a phosphorylase. Now, what enzyme are they mentioning in the question stem?
and isomerase.
Uh-huh. And that just moves stuff around. It doesn't add anything, it doesn't remove anything, right?
Mm-hmm,
So A can't be it, right?
yeah.
So, I always want my students to have more than one way to answer a question, but also one that isn't just because I don't want to say like, just know it. Like, know it now. You won't get it wrong the next time or something like that. Like, uh, so I don't like to say like, okay, you need to know that DHAP to glyceraldehyde 3 phosphate like does not involve, hydrolysis and stuff. I want you to be able to see, hey, if we're dealing with ATP at all, we're going to have these types of enzymes, but the enzyme that they mentioned isn't the type of enzyme that we're looking for, so it can't be A, right?
Mm-hmm.
Now, what about B?
The following reaction occurs very slowly. I mean, I don't think it mentions it wants to know anything about speed in the question, but Um,
So now this is an important, also high yield thing. There's, have you heard of like a thermodynamic control versus a kinetic control?
I confuse those two, but I heard of the terms.
All right. Um, so thermodynamics deals with energy of things and stability. Kinetics deals with speed. So let's say we have a regular reaction diagram. This is the reactant, this is the product, and then this I could say is the enthalpy or bond energy of the reactant. This I could say is the enthalpy or bond energy of the product. And then this is the delta H of this reaction, right? Now these are uh, simply just, energy levels of these compounds, right?
Hmm.
Now, what's this hump over here?
I mean, like the hump is like the activation energy, but in that section, it's like the transition states.
Oh, yeah, excellent.
Yeah.
So, so yep, it is the transition state and it is also the activation energy. Now, what can happen to the activation energy? Like, can we change that or anything?
If you add a catalyst, you can lower the activation energy.
All right. And if we do that, what happens?
So delta G remains the same. You just lower the activation energy that allows the reaction to happen faster,
Faster, right?
which is kinetics.
Yeah. Now, I know it's misleading because they call it activation energy, but whether we lower that or not, it doesn't change the delta H or the H of the reactant or the H of the product, right? So thermodynamic quantities would be like H, like enthalpy, bond energy, Gibbs free energy, those types of things. But kinetics deals with speed. And since the activation energy can be lowered with a catalyst and it doesn't affect the energy level of anything, but it affects speed, that means that that's a kinetic thing. So B won't be right because that's talking about the speed of something, which is a kinetic thing, and that's not, relevant to why this reaction goes forward, right? So,
Hmm.
okay. Um, oh, what about D?
The activation energy of the reaction catalyzed by the triisomerase is very small.
Hmm?
We don't know anything about the activation energy based on the
Well,
question, right?
if we did, like, let's say it's true.
Um, but it's basically saying that it's very small. That means it's very low. But I guess I'm not sure what it means by saying that it's very small.
Well,
Like, is it like to the bottom, like it's trying to change the Gibbs free energy or something? Like,
well, remember the activation energy here, and we lowered it if we added a catalyst.
yeah,
So just something like that. But it would be wrong for the same reason B is wrong because lowering the activation energy just changes the speed, right?
yeah,
We're not concerned with speed, right?
yeah.
So D would be wrong for the same reason that B would be wrong,
Okay, yeah.
right? And
Mm-hmm.
C. Now,
Interesting.
huh? Now for
Yep.
C, right?
Yeah, you do.
If I had reactant turned into product, let's say I increase the amount of product. what would happen in this reaction?
If you increase the product, and let's say it's a reversible reaction, wouldn't it just go to create more
Yep.
reactants?
And vice versa, right?
Yeah.
So if we're saying the reactant is DHAP and the product is glyceraldehyde to be phosphate, why does this proceed? Well, this could be a reason why. If we have an excess of DHAP, it should drive the reaction forward, right? Le Chatelier
Mm-hmm.
principle.
Mm-hmm.
So there you go.
Oh, okay. Oh, okay.
But you can tell it's a tricky question because look, 47%
Yeah.
chose A, right?
Mm-hmm.
And then like more than a quarter chose this activation energy thing. So, and it should make sense why people would choose things like that. Like, like if it says it's a slightly positive change, it means that you need to add energy. And so if you break ATP, you can add energy from that. So that's, I can see why people would choose things like that. But yep. All right.
Okay. This one I've done this before and it makes sense about oxidative versus phos.
Substrate level. Yep.
Yeah, something, yeah, that. Um, this one I remember doing that, right? Yeah, I was like, yeah, I knew about the hexokinase.
Uh-huh. Yeah, and
Um,
it's called glucokinase, just to let you know.
oh, glucokinase.
Yep.
Okay. Um, I think this is just something that I just need to study through, like glycolysis to know about what a rate limiting step is.
Oh
And
yeah, so that's just another way of saying irreversible step.
yeah, yeah, cytosol. Um, yeah, this one I just
So,
glucagon increases the activity of
so glucagon does what in our bodies?
glucagon. Doesn't it try to do stimulate gluconeogenesis?
Well, yeah.
Yeah,
I was gonna ask a simpler one, like it just something that increases your blood glucose.
yeah.
And so, yeah, for this question, they're really asking us, which enzyme would increase in activity, like in gluconeogenesis, for instance. So the you could also think of gluconeogenesis as like glycolysis in reverse. So glycogen synthase, what do you think that would do?
Create glycogen.
Uh-huh. And that would be when you would be storing it, storing glucose, right?
Yeah.
So if glucagon is breaking down glucose or breaking down something to make more glucose, it would be the opposite of glycogen synthase, right?
Hmm.
And phosphofructokinase, one, I believe that's in glycolysis, and I know pyruvate kinase is also in glycolysis, and also you can kind of think about it like this too, like glycolysis involves like three phosphorylation steps, or it might be four, but then that means the reverse gluconeogenesis should involve dephosphorylation, and that's what a phosphatase does in C. Yep.
Okay. To know that. I think this is just me just knowing the enzymes for these this stuff.
Yep.
Um, these are all about gluconeogenesis. We're gonna need to study that. Um, yeah, I guess these are all the like questions I have for now. I think I just need to spend time trying to study the different pathways. Um, and I see on like the schedule, I'm supposed to start doing the section bank because right now I'm just reviewing um all of the Jack Weston diagnostic. It says I'm supposed to be starting doing the section bank next week, but I think I need one more week before I start doing the section bank.
Yeah, totally. Because you're scheduled to take it in May, so we have more time. So you don't have to rush or anything. You can continue with the reviewing the content diagnostic, and then you can start that section bank. So I'll put it in for like the rest of next week so that maybe you can start it like on the 18th. I just
Mm,
change it in your calendar for that. Um but okay. So yeah, I would so
yeah.
how are you with reviewing the other two section sorry, the other two?
So like on it, I got through the psych soc one. I just need to know terms, and I did pretty well on that one in comparison to the rest. I think it's more beneficial to spend time reviewing biochem and chem phys.
Yeah, I was gonna
Chem
ask how.
phys, I did the absolute worst out of all of them.
Yep.
Which is funny because when the first time I was studying for the MCAT, it was actually the biochem that actually
Got it.
tanked.
Yeah,
But I
yeah.
think because I took a biology class, I'm just kind of a little less. And
Yep, yep. Yeah,
so I think,
that's
yeah.
usually the case. That's usually the case. So yeah, next time we meet, we should like probably go over the rest of questions from here and maybe some stuff from CP. Um, particularly like perhaps like the physics stuff. And then if you have any questions about cars, we can go over that too. But from today, I see that your cars was really good. Like, your map was really good, your highlights were good, your reasoning was good. So, yeah, you're doing an awesome job.
Okay, thank you. How much for this session? When is it?
Uh, 150, yep.
Okay, I feel like last time. Yeah.
Mhm.
And I should get my final schedule in the next few days. I just keep on waiting. 'Cause I'm trying to
Mhm.
um see why is this all caps? Sorry. And I've been like just trying to watch like YouTube videos start to familiar or like rememorize some stuff
Mhm.
that like I know I have to rememorize the amino acids because I should be getting them faster than what I've been doing. Like be able to recognize them like that.
Yeah.
But
Are you using Anki or?
I'm starting to use Anki, but like for some reason I'm much slower at recognizing there. But like I was using the amino acids app.
Mhm.
Um and that I just need to learn how to recognize the different structures in different ways and not just one way. Um that I've been trying to because I know they all look kind of different depending on what they're trying to draw or the way it looks like. But so I'm trying to get to where did Zell go? There we go. Can I put in the mail? There we go. View. It should be sending. Yeah, it says, I don't know if you got it, but it says sent on my end.
I got it.
Okay, that's good. Yeah, but like I've been using the amino acids app, like this one. I don't know if you've seen it before, but
Yeah, I would
yeah.
highly recommend though, continuing to use Anki, like especially for the other stuff, just because it's like, especially like for yeah, metabolic pathways and stuff, because there's so much you have to memorize. So I would just say that, but yeah, I would say keep up the good work. And for next time, are you gonna schedule or do you want me to?
Yeah, I'm gonna schedule. Yeah, I just need to see how the time is looking like for next week since think next week I'm officially starting to work. So we'll see.
All right,
Um
awesome. So yep, keep up the good work. Have a good weekend, and I will see you sometime next week. Okay.
okay, thank you.